Hello. In this presentation, I will tell you about gender differences in bladder wall fibrosis and compliance in spinal cord transected mice and the possible role of angiotensin signaling. I have nothing to disclose. Bladder wall fibrosis is implicated in a variety of bladder pathologies, including various forms of cystitis, outlet obstruction, and spinal cord injury. And fibrosis can decrease the compliance and capacity of the bladder and adversely affect its both storage and voiding functions. However, most of the studies using spinal cord injured mice show that they exhibit increased bladder compliance and capacity. And we hypothesized that this discrepancy may be due to the fact that female mice are protected from developing fibrosis. It may involve sex differences in regulation of angiotensin II type 1 versus angiotensin II type 2 receptors. So these are two main types of receptors for angiotensin II, and angiotensin type 1 receptor is thought to be profibrotic, while angiotensin type 2 receptor is antifibrotic. Some studies show that estrogen may either inhibit or downregulate angiotensin type 1 receptors, while testosterone can do the same thing with angiotensin type 2 receptors. So our aims were to evaluate the differences in bladder wall fibrosis in male and female mice with spinal cord injury and to characterize the involvement of angiotensin receptor signaling. The animals were transected at T8, T9 level, and at the time of surgery they were also implanted with subcutaneous osmotic pumps, slowly delivering losartan, the angiotensin type 1 receptor antagonist, or PD-123-319, the angiotensin type 2 receptor antagonist, for 4 weeks. 4 weeks post-surgery, animals were sacrificed and their bladders isolated for histology and length tension recordings. These are the typical histological sections of all the animals, stained with modified Verhoeven Giesen stain for collagen. Pink color stains collagen, yellow stains muscle, and brown nuclei. We showed that spinal cord injury results in a dramatic increase of collagen to tissue ratios only in male animals, and in females this ratio stayed at the level of sham animals. Both sham and spinal cord transected females treated with angiotensin type 2 receptor antagonists showed increased levels of collagen, while losartan decreased collagen levels in spinal cord transected males in comparison to untreated spinal cord transected males. Length tension profiles demonstrated that spinal cord injury caused a significant left shift in the length tension curve of male mice, meaning that their bladders became stiffer and were not able to relax with a stretch. Female bladder length tension profile and compliance were not affected by spinal cord injury. Losartan increased compliance and ability to relax of the spinal cord injured male mouse bladders, while angiotensin type 2 receptor antagonist dramatically decreased compliance of both sham and spinal cord injury female bladders. Thus, passive tension at optimal length, the length of maximal response to electrical field stimulation, was increased in the spinal cord injured male mice and decreased in spinal cord injury females in comparison to sham animals. Losartan decreased the passive tension of spinal cord injury male mouse bladders, while the PD drug increased it in both sham and spinal cord injury females. In conclusions, female mice may be protected against the development of bladder fibrosis due to altered expression or estrogen inhibition of angiotensin type 1 receptors, while male mice are prone to bladder fibrosis due to altered expression or testosterone inhibition of angiotensin type 2 receptors. Also, losartan, the angiotensin type 1 blocker, may have therapeutic relevance in decreasing fibrosis in adult male or postmenopausal females. Thank you.